Good morning, students. My name is V. Jyoti, lecturer in zoology. I am from Government Degree College for Women, Siddipet. Today topic is genetics, uh, introduction to animal genetics and history of genetics. Important books refer this genetics is important. Okay, next go to history of genetics. Gregor John Mendel, the father of genetics. Gregor Mendel, the or Gregor John Mendel also say, okay, genetic is the study of heredity and variation. So father of genetics, whose name? Gregor Mendel. Okay. Next, scientist year 1666. Okay, Hook scientist discovery of cells. 1831 Brown discovered of nucleus as a central feature of cells. Okay, Brown discovered by nucleus. Okay, cells new discovered by Hook. 1858 Charles Darwin, the theory of evolution through natural selection. Very important. This is a natural selection evolution. Scientist is Charles Darwin. Darwinism means natural selection. Next, in 1859 Charles Darwin published the origin of species. Very important question. It is also MS sentence bit. Okay, Charles Darwin discovery another published the origin of species on book in 1859. 1866 Gregor Mendel or Gregor John Mendel same, okay, published the result of inheritance of factors in pea plant. Very important. Inheritance of and 7 factors mainly he characterized the main 7 characters. One is the length of the stem and position of flower, color of a pod and shape of the pod and shape of the seed, color of the seed coat, color of the cotyledon. This character is called inheritance of factors in pea plant discovered Gregor Mendel. Okay, origin of species book who is published Charles Darwin. Charles Darwin. Another is natural selection is important. Robert Hooke, Robert Brown, or same Hooke is discovered. Cells Brown is discovered by central future of cell is nucleus to discover. Okay, next in the 1900 year, okay, Coral Current, Hugo De Vries and Rich von Kirchmark. Okay, Coral Cornus Hugo De Vries is 19th century discovered by Mendel's principles independently. Okay, this Coral or Hugo De Vries. Okay, important to discovered by Mendel's principle independently. And independent means is important 9:3:3:1 ratio. Okay, is hybridization, double hybridization principle identified. Okay, Hugo De Vries important in 1903 year. Okay, Johnson and WL. Johnson is coiled the term phenotype and genotype is important. In 1903, in scientist Walter Sutton and Brovey. Okay, important. 1903, Walter Sutton and Brovey is Brovey discovered by chromosomal theory of inheritance is important. Walter Sutton and Brovey is chromosomal theory of inheritance discovered. 1950 or sorry 1905, William Bateson discovered by called the genetics, discovered by which discovered this William Bateson called the term genetics. In 1905, Nettie Stephen and Edmund Wilson discovered by independently described the behavior sex chromosome like XX determined female, XY determined male is important for the Nettie Stephen and Edmund Wilson. Okay, it is called male female. It's a different behavior sex is discovered in 1905. Okay. Next scientist 1908, Correns and Bateson and Punnett discovered of genetic linkage. Very important. Correns and Bateson and Punnett discovered, this scientist discovered of gene linkage. Very important. Gene linkage, some disease also formed. In the 1910, T.H. Morgan proposed a theory of sex linked inheritance. Important. Sex linked inheritance like some Klinefelter syndrome and color blindness and some different type of disease are discovered or syndromes are discovered, followed by the gene theory including the principle of linkage. Okay, this Morgan is a T.H. Morgan, sex linked inheritance discovered this theory. 1913, Alfred Sturtevant. Okay, Alfred Sturtevant made the first genetic map of a chromosome. Alfred Sturtevant is first discovered genetic map of a chromosome. Okay, in 1913 to 1916, Bridges discovered non-disjunction as a proof of chromosome theory of inheritance. In 1927, H.J. Muller used X-ray to cause artificial gene mutation in Drosophila. Okay, it's an important question for MSc entrance. This red mark is an important question. Okay, gene linkage discovered by Correns and Bateson and Punnett. Next, H.J. Muller discovered by used to X-ray to cause artificial gene mutation in Drosophila. Okay, artificial gene mutation in Drosophila discovered by H.J. Muller. In 1928, scientist Frederick Griffith scientist, very important, discovered that heredity material from dead bacteria can be incorporated into live bacteria. So Frederick Griffith is important further, is important. Dead bacteria can be incorporated into live bacteria is important. Is Frederick Griffith. Griffith is important question. Next is the 1933 John Bratchett. Okay, showed that DNA is found in chromosomes and that RNA is present in the cytoplasm of all cells. Okay, Bean Bratchett scientists discovered important is DNA is found in chromosome and that RNA is present in the cytoplasm of all cells. Okay, they're like mRNA and tRNA and some not mRNA but rRNA is a non-genetic RNA types. Next 1936 strain 1936 scientists the strain discovered of mitotic crossing over is important. 1936 strain discovered of mitotic crossing over. 1941 Edward Ravitam and George Wells Beadle showed that gene code of proteins for proteins. Next, 1950 Erwin Chargaff is important. Chargaff rule, we know it. Very important question. This discovered a 1:1 ratio of adenine to thymine and guanine to cytosine in DNA samples. Okay, important. Okay, important. Chargaff, Edwin Chargaff means 1:1 ratio, means 1:1. How it possible to adenine paired with thymine, guanine paired with cytosine in DNA? So the samples of 1:1 is Chargaff rules, 1950. Next, this scientist in 1953 in year James Watson and Francis Crick. At Watson and Crick, we know it. James Watson Crick, Frederick Francis Crick or Watson Crick discovered double helical structure of DNA. 1953 Crick and Watson solved the three-dimensional structure of the DNA molecule. We know it DNA double helical structure and three-dimensional structure of DNA molecule also discovered Watson and Crick. Okay. Next, 1958, in same year 1953 Same 1953 is Watson Crick discovered by helical structure of DNA and three-dimensional structure of DNA molecule. Okay, 1958 Milson and Stahl semi-conservative replication of DNA. Milson, okay, Milson and Stahl discovered by semi-conservative. Semi-conservative means one is old strand, one is new strand. So it is called semi-conservative method in DNA replication. In 1958, Malthow, Michelson and Frank Stahl discovered isotope of nitrogen to prove the semi-conservative replication of DNA. Okay, next in year 1970, Hamilton, Smith and Cantwell Cox. Hamilton, Smith and Cantwell Cox discovered isolate the first restriction enzyme Hind2 is important, though Hamilton H, or do you know it? So Hamilton, Smith and Cantwell Cox is discovered by first restriction enzyme Hind2. In 1972, Powell, Bird, Herb Boyer produced the first recombinant DNA molecule. Is rDNA technology is first 1972 is Powell, Birch and Herb Boyer. In 1953, Carey B. Muller's is discovered by polymerase chain reaction is PCR, polymer chain reaction PCR. In 1983, Carey B. Muller's discovered poly chain reaction. In 1989, Alec Jeffries discovered called the term DNA fingerprint and was the first to use DNA polymerase in permitted immigration and murdered cases or crime cases. Okay, in 1989, Alec Jeffries discovered called the term DNA fingerprint and was the first to use DNA polymerase in permitted immigration and murdered cases or crime cases. Okay, it's important. Alec Jeffrey fingerprint to identify murder or crime cases. Next, this scientist in Har Govind Khurana, we know it, Indian scientist Har Govind Khurana, Indian American molecular biologist. He was awarded the Nobel Prize shared with Robert W. Hawley and Marshall Warren Nuremberg in 1968 for his work on the inter- interpretation of the genetic code and its function in protein synthesis. Har Govind Khurana is an Indian American molecular biologist. He discovered some. He also Nobel Prize and genetic code and its function in protein synthesis. What is genetics? Genetics derived from the Greek word gene, which means to become or to grow. To become or grow is called genetics. Okay, it's a Greek word. It means to become, to grow. Science characters are governed by genes. Genetics is the study of structure and composition, function or genetics. Another genetic is a science which deals with the principles of heredity and variation. It studied the inheritance of various characters from one generation to another. Heredity includes the traits or characteristics which are transmitted from generation to generation and it's therefore fixed for a particular individual. Variation are mainly of two types, heredity and environmental. Some first one is heredity variation, refer to difference in heredity traits. Such variation are found in progenesis of different parents as well as among progeny from the same parent. Heredity variations are transmitted from generation to generation. Environmental variation are these which are just due to environmental variations are temporary and have nothing to do with the previous or the next generation. Branches of genetics: animal genetics, plant genetics and microbial genetics and molecular genetics. Okay, some animal genetics, it ideals with inheritance of traits in animals. Okay, animal means traits in animals, inheritance. Plant genetics, it ideal with inheritance of characteristics in various plant species. Microbial genetics, traits in microorganisms like bacteria, virus and fungi. Next to molecular genetics, it ideals with the structure and composition, function and replication of gene. So structure and composition, function and replication of gene is molecular genetics. And next is population genetics. It deals with the frequency of genes and genotypes in a population as well as with various factors which tend to alter gene frequency in a population lead to evolutionary characters. Cytogenetics. It deals with combined common study of cytoplasm, cytology and genetics, that means study of chromosomes. Cytogenetics means the study of chromosome or cytology and genetics. Eugenics, it deals with the control of heredity disease and specifically inborn error of metabolisms. Inborn disease or inborn means after birth it metabolism. Okay. Eugenics. Next, radiation genetics. It deals with the effect of various type of radiation on chromosomes and genes. Next, eugenics. Eugenics, it deals with the application of genetics principle for the betterment of human race. Okay, betterment of human race is called eugenics. Next, Mendelian genetics. It deals with the inheritance of qualitative characteristics of or oligogenic characters with display discontinuous variation. Next, quantitative genetics. It deals with the inheritance of polygenic or quantitative characters. This branch of genetics is also known as biometrical genetics. Okay, is quantitative means it deal with inheritance of poly or quantitative characters. Quantitative characters are quantitative genetics. This branch is genetic is also known as biomaterial genetics or statistical genetics and mathematical genetics is called quantitative. Quantity means is mathematical genetics. Next, preformation theory. Okay, this theory states that a miniature human called homunculus was already present in the egg and sperm. In another word, a miniature human as preferred in the gametes. The development of gametes results only in the growth of miniature human who is already present in the egg and sperms. The theory was not acceptable because this could not be provided scientifically. Okay, it's not. So pre-Mendelian concept about heredity is not provided of preformation theory. Okay, next, theory of epigenesis. Epigenesis is discovered by scientists. Why? Which one? This theory states that egg or sperm cells do not contain miniature human. In another word, egg or sperm cells are undifferentiated. The differentiation into various organs or parts takes place only after fertilization from the zygote, resulting into development of adult tissue and organs. This concept is known as epigenesis, which is universally accepted. Wolf, okay, Wolf scientist universally accepted this theory of epigenesis. Okay, next is discovered these concepts is uh, very important with theory of epigenesis. Next, Lamarck theory of acquired characters. Lamarck theory of acquired, no, it is easy to understand. This study states that new characters, one acquired by an individual, shall pass on to its progeny. This theory was disproved by Weismann. We cut the tail of mouse for 22 successive generations and always got the baby mouse with tail. Thus, this theory was soon rejected. Okay, this Lamarck theories acquired characters is rejected. Next, theory of pangenesis. Charles Darwin. Charles Darwin. This theory states that very small, except but invisible copy of each body organ and component called gemmules are transported by the bloodstream to the sex organs. These gemmules are assembled in the gametes after fertilization. These gemmules move out to different parts of the body, resulting in the development of respective organs. This theory was also rejected because it did not have scientific basis. Next, germ plasm theory. This theory states that body tissue are of two types, with germplasm and somatoplasm. The germplasm refers to the reproductive tissue or cells which produce gametes. The somatoplasm include all other body tissue which are not related to sexual reproduction. Thus, transmission of characteristics from one generation to another takes place only through germplasm. Any change in the germplasm will lead to change in the next generation. This theory is accepted in broad science. Germplasm and somatoplasm is theory is accepted. Next, application of genetics, very important application. Genetics has important practical application in the various field. First one, taxonomy. It helps in classification of animals, plants, organism based on chromosome number. It's taxonomy. Okay. Next, veterinary. It helped to into the Importance of domestic animals. So veterinary, we know it, domestic animals, the milk production in cow and buffalo, meat production in sheep, goats and pigs, and egg production capacity in poultry having been significantly improved through the application of genetic principles. Similarity mainly improved breed of pet animals like horse, dogs, cattle, and pigeon and rabbits have been developed all over the world. Okay, important to application in veterinary and taxonomy to application to genetics. Next, medicines also used to detection or detection of heredity disease. Some heredity disease can be detected at an early stage of life. Moreover, genetic diseases can be prevented by advising future parent with the help of family pedigrees. Okay, this is called detection of hereditary disease is important of parent with the help of family pedigrees, like a pedigree diagram in the chart like form. Some is important to detect the hereditary disease like some sickle cell anemia, color blindness, some type of disease heredity to help to family pedigree is important. It's a medically use. Next to production of antibody. Special genetic strains of fungi and bacteria having been isolated to greatly increasing the yield of antibodies and another drugs. So today very important this type. Okay, production of antibody, which type special genetic strains of fungi, some this time is corona, uh, black fungi, white fungi, yellow fungi, we know it, and bacteria have been isolated to greatly increase the yield of antibiotic and another drugs. Next, evolution through both natural and artificial selection. So we know it, evolution biology, natural and artificial selection. Okay, 1 minute. Okay, in this point we observe very important question is the first one. Scientist, this important scientist. Okay, one time the father of genetics, who? Gregor Mendel, the father of genetics. Next red mark we observe in year 1666 Hooke discovered by cell. In 1859 Charles Darwin published the origin of species. Charles Darwin discovered one book, it's published origin of species and natural selection also, Charles Darwin. Next, 19 in year 1903 Johnson Campbell called the term phenotype and genotype. Johnson. Okay, next 1903 Walter Sutton and Bravey. Walter Sutton Bravey discovered by chromosomal theory of inheritance. Okay, Walton Sutton theory and Bravey discovered by chromosomal theory of inheritance. Next, 1905, in year, William Bateson called the term genetics. William Bateson, okay, called the name the genetics. Okay, next, 1908, the scientist is Lawrence Bateson and Punnett discovered by gene linkage. Gene linkage is linked is Corpen and Bateson and Punch. Next, in year 1927, H.G. Muller discovered by X-ray to use cause artificial gene mutation in Drosophila. Okay, Muller. Next is the in 1953 Watson Crick. In 1953, same Watson Crick discovered by double helical structure and three-dimensional structure of DNA molecule. Is both are same. Three-dimensional and double helical structure of DNA. In 1972, Paul Birch and Herb Boyer is discovered produces the first recombinant molecule. Okay, Herb Boyer, you know it. Birch and Boyer discovered recombinant DNA or DNA molecule. In 1983, Carey B. Muller. Okay, we know it. Carey and B. Muller is a polymer chain reaction, but we observe this. Okay, Carey B. Muller, PCR, polymer chain reaction, and next, in year 1989, Malik Jeffries discovered DNA fingerprinting. So DNA fingerprinting, how it used? First used to DNA polymerase and but is used to murder cases. So it first to Milk Jeffries. Okay, PCR is Carey and Muller's, and recombinant DNA is discovered is Birch and Boyer. Is important. So thank you for this time. Okay, next time we continue.